Start though, so welcome, a very warm welcome to another Manchester Insights event. My name is Andrew Young. I'm Senior Development Manager here at the University of Manchester with the Division of Development and Alumni Relations. So our Insights events are very popular events that we hold 3 or 4 times every year in Manchester and London, to hear from leading academics in a particular area of high-quality research endeavour, where Manchester is considered world-leading. We've sold out for this event, but for those that have missed out, I'm reliably informed we have a live streaming. We are live streaming the event, and it'll also be available on our website for you to watch again afterwards, if you like. For those that like to tweet, we have members of the audience live tweeting this evening on #UMInsights, so please do take part. Just to say, we're not anticipating a fire alarm, a fire alarm. So if the alarm sounds, please make your way to the nearest exit following the emergency exit signs. Can you also please turn off your mobile phones? So to turn to this evening's topic and introduce your speakers. At Manchester, our pioneering discoveries and interdisciplinary research are tackling some of the biggest questions facing the planet. One of the greatest challenges we face relates to how to decarbonise the energy system. Time is running out to develop solutions to avoid dangerous climate change. And tonight's topic looks at Manchester's efforts to tackle this challenge head-on. And just as Manchester was at the heart of the original industrial revolution, we are now leading the way towards a new bio-industrial revolution. Just to say that after tonight's talks, there'll be ample time for questions, so please do give that some consideration. Without further ado, I'm delighted to pass over to you to the first speaker, Professor Nigel Scrutton, who's Professor of Enzymology and Biophysical Chemistry and Director of the Manchester Institute of Biotechnology, who will chair tonight's event. Please would you extend a warm welcome to Nigel.
So thank you, Andrew, for a very kind introduction. Delighted to see a full audience here tonight, so a very warm welcome to all of you. As Andrew says, my role in the university is the director of the Manchester Institute of Biotechnology, which will try and give you a flavour today for the type of research that we do in that building. I'm joined by 2 members of the Institute. We're all also members of the School of Chemistry, Professor of Synthetic Biology, Professor Eriko Takano, and Professor David Leys, who is a Professor of Structural Biology. We all have a vested interest in new technologies that are being used and harnessed for discovering new ways of generating fuels for the future, and it's that we want to give you a flavour of today. All this work occurs here in the Manchester Institute of Biotechnology. You're all familiar, of course, with Manchester, so you'll realise that these were taken on a very rare day in Manchester when the skies are blue, but occasionally it does happen. These are a series of photographs when the building was opened way back in 2005, 2006. We've just gone through our 10th anniversary. And I think in that short period of time that we've been opened, we have been able to celebrate some major successes. And again, we'll share some of those with you later on today. So as Andrew said at the outset, of course, Manchester is the birthplace of the Industrial Revolution. And really, we want to be at the centre of the next industrial revolution. That's very much around the area of biotechnology, the so-called bio-industrial revolution. There is a burgeoning interest in how the bioeconomy will develop in the next 20, 30 years. And really, we want to put Manchester and its agenda right at the heart of that bio-industrial revolution. And in recognition of that, the university has established a series of research beacons, and industrial biotechnology is one of those beacons. And again, it's a way of getting our message out there that industrial biotechnology is very much an important part of what we're doing within the University of Manchester. So here's the MIB. If you know the geography of Manchester, you'll recognise that this is on Princes Street, opposite where the BBC used to be located, over here, which is now being redeveloped by the local council. The MIB, as I say, opened in 2006 really as the UK's first genuinely interdisciplinary research institute of its type, where chemists, engineers, biologists, computational scientists, and physicists and others can work together in a single building and really tackle some of the large sort of, if you like, grand challenges that face our society at the moment. And by putting everybody into the same melting pot, we can spark new interactions that really push on some of those grand challenges. The MIB, as it's known affectionately back in Manchester, is really pushing very hard in the area of chemicals and fuels production. And most of our staff are engaged in that type of activity. And that's been underpinned by a new and emerging science called synthetic biology that Professor Takano will tell you a little bit about, and how that's developing in the UK and helping us to fulfill our agenda in the chemicals and fuels production space. So we set out our very large sort of grand challenge areas in the MIB. They can be mapped onto three key areas: industrial biotechnology, where we're trying to create sustainable solutions to industrial chemicals manufacture, biopharmaceuticals and food security. But I guess more relevant for today's discussion is in the area of energy and biofuels, and really trying to meet the global energy challenge that we all face moving forward, and releasing our dependency, if you like, from petrochemical-based feedstocks for energy. And then our third grand challenge area is in the biomedical and healthcare areas, where we're interested in developing structures of drug targets, looking at how macromolecules in cells in the living organism can interact with each other, and trying to get a holistic view, if you like, of how all those networks of molecules talk to each other, and how we can interfere with those networks. All of this requires team-based working, so this little sort of infographic here, if you like, is supposed to show that with MIB what we're trying to do is provide leadership, national leadership, international leadership in this space, bringing all these interdisciplinary scientists together to tackle some of these major societal challenges. Now, what I'm going to do is show you just for 2 minutes one of the videos that our communications department has shown that really celebrates, if you like, industrial biotechnology here at Manchester. If I can just activate that video. This is that horrible... Oh, there we go.
We live in a society that has long been dependent on coal, oil and gas. These sources of carbon provide the raw material for manufacturing many everyday products that we take for granted. Pharmaceuticals, food and drink, materials, plastics and toiletries. But they won't last forever. The University of Manchester is at the heart of a bio-industrial revolution. We're using biological resources such as plants, algae, fungi, marine life and microorganisms to replace fossil carbon and to manufacture the chemicals and materials we depend on. We're finding solutions to some of the world's biggest challenges, from ensuring food security and affordable healthcare to making energy more efficient. We're world leaders in synthesizing and manufacturing new bio-based chemicals, bringing together the best minds to create enzymes that are crucial to industry. We've already synthesized one of the leading medicines used to tackle hepatitis C. We've engineered bacterial strains to produce flavors and fragrances. Our understanding of enzymes is helping to develop the biofuels of tomorrow. Our society is challenged with a growing and aging population, fossil carbon depletion, and climate change. To meet these challenges, chemicals manufacture in the 21st century needs to be smarter, more predictable, and more sustainable. By creating the next generation of chemicals for industry and healthcare, we're helping to revolutionize the economic, social, and environmental landscape. Global Challenges, Manchester Solutions.
So that brief video gives you an insight into what we do inside the MIB. The footage, the wonderful footage that you saw there, was taken by our communications department and really showcases some of the infrastructure that is present in the MIB. We're going to focus tonight on biofuels, and what I've done here, of course, is to capture what are the societal drivers at the moment for generating, if you like, or moving toward new generations of fuels, in particular biofuels, because, of course, we face fossil fuel depletion, environmental degradation, climate change, air pollution standards, and distributed manufacture. And all that, of course, is captured in local policy directives that are coming out of, for example, the European Union or other government bodies. And really what we need to do is to develop new, smarter biofuels, which as part of a more comprehensive fuel strategy can help fill or satisfy some of those needs. And of course, this also needs to satisfy the needs of the economic prospects of more developing areas of the world as well, perhaps in Africa, Latin America, and Asia. I need to make one definition clear to you today, that when we talk about biofuels, we talk about different generations of biofuels. First generation biofuels, which are now almost in the past, are really biofuels derived from food crops grown on arable land. Second generation are from non-food biomass. Third generation are oil-based fuels that are derived from algal growth. And fourth generation are really what we're going to focus on this evening, which is really using what we call the cell factory concept, using biology to make new fuels through fermentation-type technology and related technologies by engineering biology to form the fuels of the future. And at that point, I'm going to hand over to Eriko, who's going to tell you about synthetic biology and other aspects of the technology development.
Thanks very much, Nigel. So, welcome to the presentation. So what I'm going to tell you about is synthetic biology. So, as Nigel said, we need a new revolutionized technology. We need to get to the next industrial revolution. And we think this is going to be synthetic biology. So how did synthetic biology actually come along? Well, it actually started from the cost of DNA sequencing going down. So perhaps you remember the first human genome project, which happened in 2002. It cost about $100 million and 15 years. Now, currently, if you want your genome sequenced, you can go to the website, go to a US company. It costs about £1,000, and you'll get your result in 1 or 2 days. So you can see the cost of the DNA sequencing has really decreased, and this has really been enabling us to get a lot of genetic information. At the same time, we've been able to start to synthesize genes. So at the beginning, it was very short pieces of DNA, but now we can make long genes, long stretches of DNA. And again, the cost of these synthesizing DNA has gone down a lot. So if you think about 10 years ago, we were reading DNA. Now we can actually start writing DNA. And so doing this, so synthetic biology, we're trying to bring engineering into biology. So you can see an E. coli cell kind of looking like a computer, because we want to make parts which we can put together like Lego bricks and make it more predictable. So the advances in biological understanding, as well as advances in smart data interactive technologies, has really driven us into the domain of synthetic biology. And one thing that's really important for this synthetic biology is that we have this design, build, test, analyze, or learn cycle. So we're doing this iterative cycle and bringing multidisciplinary people together, from computing to biology, chemistry, biochemistry, even social sciences people together to actually do this cycle. And basically what we're trying to do is speed up and be more predictable in doing the bio design. So if I give you some examples of what synthetic biology is, we can think of some things like total genome synthesis. So this is a Mycoplasma mycodes. This is the first chemical synthesis of genome. But recently, in 2017, we had Saccharomyces cerevisiae, 4 of those chromosomes being completely, totally synthesized. But if you think of another way of doing it, we can think about using actually natural compounds, what nature has given us, and to redesign that. So here's 2 examples. One is about Artemisinin, which is an anti-malarial drug. Sweet wormwood is the plant that actually makes this compound, but because it's made from plants and growth is quite difficult. Purifying is quite difficult. So one of the researchers in UC Berkeley, what they did was to take enzymes from plants and put them into Saccharomyces cerevisiae, and now they can actually produce this compound in yeast. This has, in fact, been commercialized, and Sanofi is not really selling it, but giving out for free to the developing countries. Another compound we can look at is vanilla. So the main compound of vanilla is vanillin, is this compound here. And it's usually produced by this orchid, but only 1% is really actually produced by this orchid. And everything else that you eat in your ice cream is usually produced from total chemical synthesis of coal tar. And so there's another company who's decided to take this enzymes from this plant orchid and put it into Saccharomyces cerevisiae. And now we can make vanillin in Saccharomyces in the yeast. And this is also a commercial compound. It's been in production about 2 years now. And so these two compounds are probably one of the first commercial products that's been using synthetic biology to produce them. Now, of course, the research councils also acknowledge the fact that synthetic biology is a very important emerging technology. So you can see here, there's a lot of funding activities already starting from 2007, and a lot of policy activities as well. But in fact, one of the biggest investment from the research councils have been 2013-14, where there was synthetic biology research centers, about 6 of them funded within the whole of the UK. Now, each of these centers have different topics. For example, Edinburgh is focusing more on synthetic biology of mammalian cells. For example, Cambridge for plants. And of course, we in Manchester have been awarded our Synthetic Biology Research Centre as well. And we're focusing mainly on fine and specialty chemical production, and we'll come back to this in a bit. And you can see from this chart here, there's more than 300 million of public investment to establish this UK-wide network in synthetic biology. And of course, if the government is funding so much into research, they would like to see something coming back into the economy. And if you look at these startups for the last 16 years, there's about 150 startup companies. Of course, not everything works, but more than 70% is still working. There's a start companies still working. And you can see that there's more than 600 million they've raised for these startup companies. Now, if you look at the startup company activity across the UK, the majority, as you can see, is south. It's based here in London. So northwest is this green line here. We're not actually there yet compared to London, but we're working hard to see if we can get this close to the London figures. So what kind of companies are there for startups? Well, you can see that there's quite a few different ones. There's those startups that are working on technology, but also on industrial biotech, chemical production, and agri-tech as well. Now, if we come back to synthetic biology in Manchester, our Symbiochem, so our strategy is to lead innovation in bio-based chemical and industrial biotechnology sectors. Basically, it means that we're working on trying to produce fine and specialty chemicals, which includes biofuels. And our strategy is that we want to build teams of interdisciplinary scientists and to have industrial partners as well. So our center is directed by Nigel Scrutton, Nick Turner, and myself. And you can see here, our strength is that we have so many PIs with different expertise. David's here as one of the co-PIs as well. And we're also very fortunate to have a team of synthetic biology experimental team, which again has different expertise in the design, build, and test cycle. We've also been developing technologies, platforms in technology. So for example, in the design area, this is computational, in silico modeling and softwares. We have a lot of state-of-art analytical equipment in the test area. We also have quite a few high-throughput robotic platforms. And because we couldn't invite you to Manchester, we've brought you the lab. And hopefully we can show you if we go... Sorry. this is the high-throughput robotics in the lab. And you can see that this is one of the most important technologies in synthetic biology in that we can do high throughput. So this robotics we can use for transforming E. coli, doing enzyme reactions, transformations, and so forth. And so if you keep on running this overnight, it can do much more than a PhD student can do. So this is one of the most important technologies. Let's see how long this will take. You can see 96 well plates. There's quite a few of them. In fact, you can't see all of it. This is the main picture of the robot. We have 3 of these robots, in fact, and they're running constantly, working on different things. Okay, so that's the end. So, okay. So, of course, we've been running this Synthetic Biology Research Center for about 2.5 years now, and we're in the middle of our 5-year project period. And we have some very early outputs. So you can see that we've been, sorry about that. Yeah, thank you. So we have more than 200 peer-reviewed publications in high impact factor journals as well. We've been training lots of PhD students for the next generation of biological engineers. We also have co-developed projects with many industrial partners. You can see some of the industry that we collaborate with. And we've also patented quite a few of our projects as well. And for this topic today, you can see that we have some new synthetic biology routes to biofuels being patented and working together with big industry. Last but not least, we've been spinning out some technology, and C3 Biotechnologies has been founded in 2015, and Nigel will be talking more about this later on. And with that, I can hand on to David.
Thank you. Okay, again, warm welcome to everyone. Thank you for coming to today's talk. So I'll be talking, as the title says, microbial platforms and enzymes. And what I mean with that is that I think we traditionally have always sort of worked on organisms, components, and metabolism, and these are just pretty pictures, ultimately to understand the chemistry that underpins life. We're now, as Eriko said, we can read and write DNA. You can do these sort of ideas of making your robotic cells, not with chips in them, but actually reconstituted biological components. And you can maybe start thinking about somehow having these elements make chemistry that you would be interested in. And so that's really today's topic. Somehow we want to get biomass to make new molecules for us, or molecules that we're really interested in. And I think most of you might well think, well, maybe there is something here that's quite difficult. We haven't really done this yet in any great scale. And I always talk about, is there not some sort of chemical incompatibility between biomass on the right-hand side and oil on the left-hand side? And you would think that perhaps there is a problem there. And so when I depict molecules here, and I don't want to lecture you on chemistry, but effectively, what is the difference for those of you who don't like chemistry? The take-home difference is that here there are none of these pesky oxygens and nitrogens and sulfur which so decorate biomass, which you can see on the right-hand side. So effectively, the challenge for us as chemists is really take your molecules on the right-hand side and convert them to something that looks like here, which is effectively fossil fuel. And we haven't been very good at that, and we are clearly working on this. Now, you might say, well, there are biofuels. Nigel mentioned first generation, second generation, and so on. And so we obviously have things like ethanol, which most of us probably would think that this may be better for drinking. And you know, maybe not to such a large extent as blend into a car, but this is obviously some technology that we've been pretty good at well before the MIB was established. And so this is something that drove that first and second generation biofuel development. Nigel also mentioned that if you take fatty acids, you can convert them. You can't quite remove those pesky oxygens, but you can make something. This is called biodiesel, or fatty acid methyl esters for the chemists among the audience. And these effectively can again be used as a sort of blend-in fuel. And the blend-in concept is one where you sort of want to use the present-day technology but make it more acceptable to use biofuels. And one of the things that lots of people talk about within the biofuel field is the need to get drop-in biofuels. And with that, what we mean is that it's all very well thinking about new molecules that might mimic things that you can see on the left-hand side, but if they're not quite in tune with the existing technology, it will be quite difficult to get buy-in. I think most of us will have cars. If I come up with a biofuel that isn't actually going to run in your car, you're not going to buy it off me. So we bought into technology a long time ago. In a way, we're now stuck with it, a bit like, I guess, the London system is stuck with the tube system. You can't really change it very easily. That's how it works. And what we're trying to do at MIB, and lots of other people are trying to do this all over the world, is somehow think a bit more imaginative and go, are there no ways that we can really make something that is truly indistinguishable from oil? Maybe a bit like Eriko was saying, think about this vanillin from the orchid. Can we make something that tastes the same, is exactly the same, but is derived from biomass rather than from fossil fuel? And it turns out that actually biology can do this. And when you look at this little picture here, you can see an algae, and this algae under very special conditions will actually turn into an oil-making machine. The oil you can actually see on the right-hand side, it actually spits out oil. Now this wonderful organism is called Botrycoccus, and it would be very nice if it were to do this at large scale. It turns out, as always, that it doesn't. So the problem with this organism is that it's quite low yield, so the biomass is very difficult to accumulate because it does this as a sort of way of escaping a limitation in terms of nitrogen. So it gets lots of sunlight. If it can't grow, then it will somehow use this light to just drive the conversion of carbon dioxide into oil. It's quite difficult to culture, it needs light, and the biochemistry of this is still quite unknown. So we're a bit stuck then, aren't we? The other problem, I'm going to talk about a few problems here before I come up with some solutions, is that biofuels isn't really one thing, similarly as fuels isn't one thing. So I think one of the key things to remember is we are obviously looking at various ways of solving the transport problem. Biofuels isn't a single solution. We all know about Tesla and other types of changes. But there are certain things where, for example, batteries are going to be very difficult. And one of these is, for example, jet fuel. I think it's quite clear that we're not going to stop flying, but we're not really going to do this with huge amounts of batteries. And so, again, there are certain types of fuels where alternative technology is a little bit more limited. And so this is a concept that Eriko was already alluding to, is that we can read and we can write DNA. We know so many genomes of so many organisms. We know some can do certain components. And all of these components are somehow linked into something that we call central metabolism. Our metabolism is actually remarkably similar to plants and microbes. That idea preceded the sort of diversion into the different organisms. So it would mean that if we can take some of these components that are really responsible for making biofuels and we place them within the context of an organism that we can handle a little bit better, for example, that doesn't need light or that we can make biomass of, that we could put two and two together and make some of these molecules that don't contain our oxygen and our nitrogen. So you can see here that the idea is to take biomass, bring it to a fermentative type plant, and then convert it as much as we can to the corresponding biofuel. Now, besides what molecule would we like to make, there is also what organism would we like to use. And again, the reason why so many people are trying to develop this and there are so many different approaches is because there are so many variables. So if you think about which organism would I want to use in these silos, you can think about an organism that Eriko already mentioned, E. coli. That's our standard workhorse in the laboratory. It's not something that industry would like to use. And so you can see here that there are model organisms, the E. coli one. There are organisms that actually already grow on biomass, but they obviously grow for themselves. They don't make oil for us. So we have to try and tease out an organism that can grow on biomass, but make these biofuels. And so this is a very busy slide. Again, it's not really me to try and go, let's talk about all these molecules. That's from the, my students will end up suffering this. What I just wanted to show here is that, again, like there are many different organisms, and we're not quite sure which is the best one. And it might well turn out that for different countries, different environment, different biomass, there will be different solutions. Yeah, we can no longer think that there will be one global solution that will just replace fossil oils. And what you can see here is that besides these different organisms, there are also different pathways and different enzymes. And I myself am a structural biologist, as I was introduced, so we like to tinker with enzymes, determine the structure, figuring out what they do really. Because that's essential for somebody who wants to do engineering. You can't really do engineering unless you understand the bits that you're dealing with. Alternatively, you might well get some, rather odd consequences. And so as a just brief example to show you where things can come from very uncommon areas, is that if you look at spoilage yeast, and when you think, what do you mean with spoilage yeast? Well, these are yeasts that will grow on mother's, homemade jam, unfortunately not necessarily so quickly on industrial-made jam, but eventually they will do so. And why do they even grow on industrial jams? If you look at industrially made food, quite often it will contain these type of molecules. Again, there is some pesky oxygen there. And these organisms, if they can grow on this, they will contain 2 proteins, and they will make something like this. This looks quite a lot like a biofuel, but nobody quite knew how it did so. The second thing is actually that it makes it smell like kerosene. So I've been told if you take your jam jar and you keep it closed for quite a lot of time, you have a little whiff. I don't say you have to do this now, but that there is a kerosene-like smell. And this was something that attracted us to this type of thing. Now, I don't want to go into a huge amount of detail, but in short, what we found is, looking at the structure of this enzyme, we found something that was quite unknown to biochemistry in general, so a bit of serendipity. We went to look out for something that can make biofuels. So we found a whole new series of biochemistry occurring, and that is now something that hopefully, not tomorrow, but in a couple of, let's say, years, I'm hoping, will give rise to new biofuel production. And we're going to pass on to Nigel, who is going to talk about C3 as a more applied pathway.
Okay, thanks, David. So in the closing part of the talk, I'd like to talk a little bit about another biofuel that we focused on quite a lot in recent years in Manchester, and that's a biological source of the carbon-3 gas propane. So gaseous hydrocarbons can be made by biology, and what we've been doing is really trying to understand the basic metabolic pathways that we can engineer to make a gaseous hydrocarbon, such as biopropane, to replace the current use of propane derived from petrochemical sources that is clearly quite widespread in terms of our energy demand. So, of course, the benefits is that biopropane as a bio-based production would give us a clean product from a wide range of available feedstocks. They may be waste feedstocks, it may be industrial waste, it may be plant waste that we can use and grow bacteria through fermentation and use those bacteria then to generate biopropane as a product of that fermentation. By doing this in fermentation type environment, of course, we then don't have to depend very much on the use of land where we might want to grow crops for food or other reasons. So that's a major benefit. There's reduced toxicity to biology. David talked about how the chemicals we find in oil are not always terribly compatible with biology. And if we produce them inside the cell, then of course that's going to be a major engineering problem. But the beauty of working with gaseous fuels is that as you're fermenting your bacteria, they come off and they collect in the headspace above the bacteria, and you can very easily remove them through liquefaction and pressurization and other types of processes. So the current drivers for trying to develop a platform for making biopropane is that, first of all, propane is the third most widely used motor fuel on the planet. About 20 million tons are used a year. It provides heat and energy for 14 million homes worldwide annually. It's got an existing global market for stationary and mobile applications. And as I mentioned previously, it's easily separated by liquefaction from the biotechnological process that's making it. So toxicity is much less of an issue when you want to make biopropane. So again, as David's mentioned, what we do here is to think about component enzymes, catalysts that are present in nature. We think about wonderful ways of bringing all those enzymes together to make new metabolic pathways that don't naturally form or are not naturally found in nature. But by bringing those combinations together, we might be able to then imagine a metabolic sequence that can take us from a central part of a bacteria's central metabolism all the way down and get a chemical conversion that eventually will generate propane gas. So that's the challenge we set ourselves a number of years ago. We designed a conceptual pathway for the production of propane in E. coli. We identified each of the individual enzymes in this metabolic sequence, and then using the techniques that Eriko talked about, where we can actually write the DNA for these genes. That's exactly what we did in the lab. We spliced them together, introduced them into a bacterial host, in this case our workhorse Escherichia coli, and then we went round this design-build test cycle a number of times to find optimal strains that could, in principle, make propane gas. And we do this in a combinatorial way. So there are lots of different components here. We can arrange these different components in very many different ways. And this is why we need this robotics infrastructure that we talked about in the center, because we might want to make 1,000 different strains with different combinations of genes put together, and then to screen those in rapid sort of automated assays to find the best propane producing strains. So that's exactly what we've done. And hey, presto, when we do that, we can actually ferment bacteria, E. coli, and in doing so, we can actually begin for the first time to see propane production from a biological organism. So this is really quite a potent milestone, I think, or an important milestone in developing a biological route to one of the most widely used gaseous hydrocarbon fuels that we currently have. And this is a drop-in fuel. Again, as David mentioned, we have a lot of infrastructure around for exploiting propane in our current societal infrastructures. And so if we can generate a scaled process whereby we can make propane gas in this way, then this could be used as a genuine replacement for conventional petrochemical-derived propane gas. So we're academics, so we do things in a rather Heath Robinson way. And this is a fermenter that, in fact, we designed with one of my industrial colleagues in C3 Biotechnology. This is actually sat on the quayside on Guernsey Harbour. Why do you want to go to Guernsey? Not because of any tax reasons, but because there's in fact a biodiesel plant just behind here that generates a lot of biodiesel waste, which we can use free of charge as a feedstock to grow our bacteria. And if we do that from here, here's our biodiesel waste. We feed that into this big fermenter here, and we can then collect off the propane gas using this liquefaction type process shown here. So I'm not an engineer, but I'm collaborating with engineers to develop a process that allows us to ferment these bacteria in this sort of steel-clad, rather, well, it's not a very high-tech fermenter, but hopefully it's the first step, baby steps toward developing a process. So we have a 400-litre fermenter. Our aim is to detect propane gas. And this is a world first. This is the first time I've shown this video to anybody on the planet, so you're very privileged. But this here, if I can get the video rolling, is actually biopropane, biologically produced propane gas burning from a cylinder that's collected from a bioreactor of this type. It's not pure biopropane, I have to be honest with you. There are other gases in there that also combust. There's a little bit of hydrogen, there's a little bit of other things that will also burn, but there's a significant amount of biopropane. So this, I think, is the first for Manchester. It's the first for synthetic biology, in that we're beginning to generate combustible fuels using biology from waste feedstocks. And this is now being developed as a process in our Manchester spin-out, C3 Biotechnologies, which is an MIB spin-out. And really, that's quite a rapid progression from the initial opening, I think, of our SynBio Centre, which only opened 2.5 years ago, to forming a spin-out here. And we're now looking to actively commercialise this process with the university's help. So there's a prospect for scalable, sustainable and cost-effective routes to propane manufacture, interfacing with widely available feedstocks, biomass, glycerin from biodiesel, industrial CO waste and other types of feedstock. It's a bolt-on to existing processes. It's a truly drop-in type technology. and of course will give us less than socio-economic and environmental constraints. It's carbon-efficient production platform. It reduces the CO2 footprint, and this can be made in a distributed way. So we could use this technology not just here in the UK, we could use it in Africa, where propane, for example, is a major fuel for domestic cooking and heating. We can make it locally there on site without shipping in propane from petrochemical sources in large tanks. So there's a real opportunity here that we want to see develop. So that gives you a flavour of where we're going. Andrew, I understand we are slightly over time, but at that point I'm going to say thank you for hearing the presentation. We're very happy now to engage in a Q&A session. If people have got questions they want to ask the panel, then we'll do our very best to answer them in the best way that we can.
Thank you. Can we show our appreciation for Nigel? So we have time for questions. Who would like to go first?
Just a question.
Can we wait for the microphone?
Okay.
Okay.
Thank you. Hello? Yeah. Thank you for that really interesting presentation. I just wanted to know the efficiency of biofuels. Have you compared to fossil fuels?
We're a long way, if we're honest, at this stage in terms of looking at efficiency. We are still looking at these processes at a lab scale type level. What I showed you here on the key side of Guernsey is perhaps the most scaled up version we've done, and it is a pretty Heath Robinson sort of contraption. We are obviously testing out the system to see how much gas is made. We haven't done the hard numbers yet in terms of efficiency. Whether any of this is scalable has clearly got to be developed in the future in collaboration with our chemical engineering community friends and others who are much more informed about that type of aspect of the problem than perhaps we are as humble genetic or synthetic biology or engineering biology individuals. There is a long way to go, so I don't want to sell this as a sort of, we've got a solution for tomorrow, but we might have a solution that can contribute to a more comprehensive energy policy in different parts of the world, perhaps sooner than maybe we thought maybe a few years ago.
Can we take the question here, please? This gentleman.
Thank you. Charles Hughes. Thank you so much. Fascinating, interesting. I think the only point I might disagree with is that the statistics indicate that Manchester had more hours of sunshine over the past 12 months than did London. We will not talk about precipitation. Two questions. Palm oil has hit the headlines recently, and there seem to be some people pushing for a banning of palm oil for all sorts of different reasons, orangutans, forest and so on and so forth, which would significantly damage the economies of Malaysia and particularly Indonesia. I'd be interested in your views on that position. Secondly, you're doing a lot of work with engineering enzymes, developing DNA, and doing all sorts of wonderful synthesis. Do you run into any significant ethical issues? And if so, how do you handle those?
So maybe I can take up on the first and second question. So it's interesting that you talk about palm oil because actually I have one slide. Nigel, how do you just go and show?
In this deck?
No, it's in the separate slide. I think we put it on your... Sorry, I'm not a Mac person.
You keep talking.
Okay, so we have actually worked on palm oil, exactly what you said, and we did this as a part of a project of undergraduate students, which we call IGEM. There's a slide up now. So IGEM stands for International Genetically Engineered Machine Competition. It's run by undergraduates, and they give up their summer holidays to work on projects using standard parts. And there's about 300 different international teams coming from all over the world, and they come all to Boston in around October time and compete with different projects. Now, the palm oil that you were talking about is this 2013 Manchester team. In fact, our team has worked on this palm oil. So what their idea was, exactly as you say, palm oil there's lots of use for palm oil, but we were cutting down all the rainforests to put the palm oil. So can we not make this in E. coli? Now, that was the experimental part of the project, but the team also did some economic studies to see if it was really worth doing this production in E. coli. What would happen to the Malaysian farmers with all the palm oil trees coming off, and okay, they have no more living, to make living. So in the end, one of the students was an economics student, and he decided that, in fact, it wasn't actually worth putting all the palm oil production into E. coli because of this social problem for the Malaysian farmers. And instead, to do this parallel, have some palm oil being produced by the Malaysian farmers, but at the same time have E. coli being producing some palm oil as well, so that you're not going to devastate all of the rainforest. So it was a very interesting idea in the end that the students came up. So your second question, so in that sense, yes, I think palm oil is a big problem, and you can see already from the social, societal problems that you can't just bang on head into this new technology, and you really have to think about a bigger picture. And of course, we do come into ethical problems. Ethical problems is different in different countries. For example, in the States, GM food, soybean, is perfectly fine, while in Europe, it's not something that's tolerated. So the way we try and work with this is, in fact, in our center, we have a special team of people who are working on responsible research innovation. And they're people with background from business schools and social sciences. And they, in fact, talk with the publics, talk with us as well, to see how is it that we can be responsibly making all these new emerging technologies. So, of course, it's not a straight answer in saying, okay, we have one ethical problem, so how do we solve that? I think it's all something that you have to think about with each different topic. For example, with the palm oil could be one example, perhaps thinking about cloning humans. You saw already in the news that monkeys can be cloned now. It's kind of synthetic biology, but not really. But again, these kind of ethical things, we have to think about it. But there's no one big straight answer that I can say, okay, this is the solution for all social and ethical problems. We have to think it one at a time, and to engage and to be able to speak clearly with the public.
Thank you. Very comprehensive answer. Just one final follow-up. The corollary to that is that surely everything that you're doing and the people who are looking over your shoulder ethically ought to be transparent and public. And the feeling a lot of people get a lot of the time is that it's not.
I agree. So I think this is one of the things that we really have to work on, is to be transparent. What we do has to be transparent. And I think in our center as well, we are being very transparent in everything what we do.
So I would add to that and say that the synthetic biology community in the UK has been very proactive about ensuring that this RRI, responsible research innovation, is embedded in all academic activities from the outset of a particular project. So that engagement with the community, whoever that may be, policy people, people on the street who are interested in the technology for reasons but don't perhaps understand the technology in a way that they need to. That engagement is happening at a very early stage. And it is about, you're right, about being transparent and open and not being sort of branded as crazy scientists, in a laboratory with four walls and trying to do things in absence of having those discussions.
And I think we learned our lesson from GMO. Perhaps some of you remember the genetic tomato that was sold in Sainsbury's as like chopped, tomato cans. I think we've learned our lesson with GMO, so we must be transparent.
Hi, Alex Wolf, a Manchester alumni, and now look after South East and London for Certus Energy, the UK's largest fuel distributor. I find the research fascinating. I think proud to be attached to Manchester. One of the interesting things we have from our customer side, so whether it's national government, local government, big industrial users is not just carbon reduction, but it's NOx reduction and particulate reduction. And I'm just interested on those two areas, what potentially fourth generation biofuels might be able to help the market with.
So I could speak to that, and you're absolutely right. Diesel, of course, and particulates and NOx has been in the news for countless articles and is a major problem. So if I could just pick up again on propane as an LPG type component, propane, butane, other types of hydrocarbon that feed into that particular fuel. There, I think it's well known that NOx production and particulate production is much less pronounced than it would be sort of burning a diesel fuel. Propane is also not a greenhouse gas. So we do already make hydrocarbon gas in this country. We make biomethane, for example, through anaerobic digestion type technologies. Methane is a major problem because methane, if it leaks out into the environment through a failure in engineering or whatever, is 30 times more potent as a greenhouse gas than propane is. So one could imagine maybe developing a biopropane technology that could essentially begin to replace what we're doing in the biogas industry, or at least adding to that initially, and then transitioning into a more propane-based production process. And what are the benefits of that? Well, the benefits I've just alluded to are lack of greenhouse gas impact. Propane, of course, you can transport very easily through liquefaction. You can't do that with methane and other biogas constituents. So there are lots of benefits, I think. Of course, propane is more calorific. You get more bang for your buck, as it were, if you burn it than you do if you burn methane. So there are lots of gains to be had here, and it's not just on the quality of the air we breathe. It's on climate change. It's on renewable production, sustainable manufacturing, so on and so forth.
Okay, there's questions. This gentleman here.
Yes, good evening. Thank you for that very interesting talk. But I do feel a little bit disappointed because the title of the talk suggested new energy for the future. And a lot of my friends here are from the oil and gas industry. We're thinking about tidal power, we're thinking about solar power, and we're thinking about all those potential. Now, okay, you've got a biofuels interest, which is fascinating. And I've learned a lot tonight. My question is, the third speaker, David, made a point of saying, algae, it's not going to work. It has problems. And you also mentioned that this wonderful vanilla orchid is also not going to ever be sustainable in volume. So my question is, when do you actually say, okay, let's ditch these? Propane works, let's move on, do something else.
Well, I think
save some money. Come on, give us a tax back.
Yeah. Well, I think propane works. It's a work in progress. I guess what you mean with algae don't work is the. Well, if you look at the whole cycle of this, is thinking about light, and if you want to make so many barrels of oil as a replacement per day, because that's what we're using millions of barrels. I think this is the main problem, that we're never quite sure as to how much we will ever be able to scale up biofuels to a level that it will actually make a dent as a replacement. I mean, so like you pointed out, alternative energies. I mean, BP, I think, recently released this idea that actually there is still more. They're surprised by how much wind and alternative energy is coming within the UK. Biofuels have something to play in there, but the question really is how much biomass can we convert and at what efficiency? I don't think anyone can give you an answer there that's really based on a huge amount of measurements just yet. It's early days. I think the reason why we're doing this research as well is to know about the capabilities. I mean, I think this is coming to both the ethical points as well as, synthetic biology is a new area. It's a revolution by the fact that it opens up new avenues, and the question is, what will those bring us? I think within the biofuels, the problem is really how much biomass can we convert. And what biomass will that be?
I could add to that and say that, I think we need to think of a comprehensive energy strategy and not a sort of single solution strategy. So, okay, you're right, and I know the industry you're coming from, you're pushing for maybe a replacement technology that would displace what we're doing at the moment. But I would say that biofuels is a component of solar, wind, tidal, whatever it might be, all pushing us into a new frontier in terms of energy provision. And we will differentiate those technologies into different sectors where they're most applicable. So I've already talked about propane and the AD industry and the biogas industry. There are 130-odd biogas manufacturing plants in the UK alone. So if we can add bolt-on reactors to an AD process and improve on that process, that surely is a win-win. Okay, it might be a relatively small sector compared to our requirements for transport fuels or whatever, but it's a significant commercial opportunity. And I would say Rome wasn't built in a day, and you need to start with these technologies and develop them. And I think it's absolutely the wrong idea to pull the rug just because algal biofuels perhaps isn't realizing its full potential after 5 years, or maybe we learn from that and we move in a slightly different direction, but we should still begin to think about more creative ways of creating biofuels, which is the fourth generation period, and I'm sure there'll be a fifth generation that happens as well in the future. So let's not give up. I think, we owe it to our children, our grandchildren to make a cleaner world, and this is one way of doing it.
Still more questions? Okay, gentleman here.
Thank you. Simon Green, Paper Science, a rather long time ago. When I look in my recycling bin, it's distressing to see the amount of paper that's really not suitable for recycling. And for that matter, you look in and look at the plastic, it may not be suitable for recycling, leaving aside economic changes and what the Chinese will take and what they won't and so on. Do you anticipate using feedstock such as paper, which after all is originally plants, and plastic, which certainly isn't, as feedstock in these technologies in some years to come?
Why not?
I think that would be nice. I think, again, yes, good idea. Yeah. I think that the difficulty is, again, maybe talking as an enzymologist. I mean, take your plastics, for example. We created them not to be damaged by water, not to be damaged by light. And so now asking the opposite question, can we on our terms digest plastic? And actually, I think, I can't remember the research, but there was somebody in Japan who found that there are bacteria who will extremely slowly grow on something like PVC or a related plastic. Then becomes the question, let's assume we can make a bacteria who eats plastic really well. How are we gonna, just make it eat what's in your bin, but not what you didn't put in your bin?
You could put the enzyme in the plastic already and make the plastic.
Yeah. So it's one of these things we do have, I guess, because this is how our society grew. It's only recently that we've come to think about limitations and now thinking about solutions. And I think, as Nigel said, 5th, 6th, 10th generation, the more molecules we can convert back into biofuels, the better. And that's one of the reasons why there won't be one feedstock and there won't be one golden enzyme that will eat all, and there won't be one bacteria. And this means that somehow our progress, if you measure this, is not necessarily incremental, but is doing multiple different things at different times. Certain people say, well, let's try with this biomass. As Nigel says, they're focusing on propane. That's not going to fly a plane, but then that's again a different thing. So that I think is the problem in this field. Too many molecules going in, and at the same time too many molecules that we want to get out. And we all try to do this simultaneously because they're all a problem.
How about contaminated cellulose then? I mean, that's
I think they are.
by a lot of things.
Well, yeah, I think we can learn a lot from what happens in biomass cellulose degradation at the moment. One of getting a pure biological process to degrade such large, difficult feedstocks is not straightforward. But hybrid technologies which are mixing sort of more chemical approaches to degradation coupled then to biochemical or biological approaches is a very major area of research activity at present. And we do have evidence that that type of approach is reasonably successful under certain circumstances. There's a lot still to be done. But taking lignocellulosic waste from many agricultural or industrial processes, I think the reality is we will have a feedstock there for the future that could be degraded. It just requires a little bit more effort to come up with these hybrid technologies to realise that.
Okay, we have time for one more question.
At the very back.
This lady at the back.
Thank you. Mel McMahon, I'm currently a member of the Academic and Alumni Advisory Board, but I'm speaking as an economics graduate. And I was a bit embarrassed about that earlier until I heard that you'd taken advice from an economics student. So I feel more comfortable about being here. Now, I don't know chemistry. I was thrown out of chemistry in grammar school. I know nothing about chemistry, but I'm interested in this subject. But it seemed to me from what you were saying tonight that you are currently researching in looking at new ways of producing propane gas, oil, etc., etc. Is any of your research focused on finding something completely different to replace any of these standard fuels?
I think that is that drop-in argument again. So there have been publications, not from our group or groups, but where people have come up with different types of molecules and looked at the combustible properties. And I think then the issue is that if you have to change anything about the combustion engine, that from an economics point of view, I mean, it's the same when we talk to companies, they go, we have a multi-billion-pound infrastructure. This was Nigel's point as well. With propane, you can feed into that infrastructure. Trying to get society as a whole to start using a new type of fuel, which would require major change in terms of infrastructure that exists, it doesn't seem to be, well, I have never found anyone who was from the industry who was saying, well, that's great, let's forget about the old and go with the new. Obviously, as the problem becomes more pressing, there might be more of a willingness to do so, but at the moment, all of the, let's say, industry-linked funding is for these drop-in fuels, making molecules we already pretty well know how to work with.
Having said that, if it's not for biofuels, if it's for chemicals, just fine specialty chemicals, for example, flavors, fragrances, even drugs, we are trying to make new chemical structures as well. So it would become a new, non-natural compound, if that's the right word for it. So in chemistry, in terms of these kind of special chemical compounds, we are doing it. For biofuels, it's a little bit of a different, I think, there's other restrictions that might stop it from trying to get these new compounds. But in other areas, there are activities trying to find new compounds.
So I would say, although the three of us are in a school of chemistry, none of us are chemists. And that reflects the background of where we come from. But we are aware, of course, that for creating certain types of molecules using synthetic chemistry, the challenges are extremely difficult. What biology can do is solve some of those problems that synthetic chemistry can't. So you can begin to build new molecules in a way that traditional synthetic organic chemists will struggle with. Now, that's great if you're in the drug discovery industry, for example. You can begin to design new types of molecule that might have new bioactive properties that might be the drug of the future. Or you might be able to make new advanced materials that can have application in society in different ways, using a biological approach that chemists can't do. So I think this underpinning synthetic biology technology is really very powerful. Not just for fuels, but for pharmaceuticals, for advanced materials, and other sectors of life as well. So, certainly worth investing in.
Great, thank you very much. Just in closing, thank you all very much for attending tonight. We hope you've enjoyed it. If you have, please do consider coming to our next Alumni Insights event, which is in another area of pioneering science you might have heard of, graphene. That's in Manchester, I think, though. So that's in June. So please, if you can make it up to Manchester, you'd be very welcome. Just to say that Please do stay around afterwards. Continue the discussions over drinks and canapes. We'd love you to do that. Thank you so much for attending. Thank you for all your support and involvement with the University of Manchester. We really do appreciate it and welcome your involvement and ongoing support. So thank you very much. Eriko wants to say one more thing.
I'm really sorry. So this iGEM project that we do every year is run by undergraduate students, but we really don't have a lot of finances. So we are desperately looking for sponsors so that they can actually travel to Boston. So if any of you are interested, please do contact me and we can have lots more discussions. They're great undergraduate students, so please do support them. Thank you.
That's where like a direct appeal for support. Thank you, Eriko. Yeah, please do join us. Just remains to me to say thank you to our three speakers, Nigel, Eriko, and David. Please show your appreciation. Thank you very much.